Hello, bio students. Today we're going to continue our molecular biology unit, and we're going to start with lesson number 7, mutations. So your note taker looks like this. It's 2 pages, so make sure you have it out and you follow along, take notes while you watch the video lesson. All right, let's get started. Okay, so list 3 ways mutation provide genetic diversity. So we know that if you're a genetically diverse population, you're more likely to survive changing environments. So that means that if a particular environment has a pathogen that makes one person sick and you're all genetically identical, then it's very likely that that pathogen is going to wipe out an entire population. But if you're genetically diverse, then you're more likely to survive. When chromosomes recombine, mutations provide for that genetic diversity. And sometimes those new combinations have little effect. Some can produce organisms that are better suited to their environments. So that would be considered to be a beneficial mutation. And others can be deleterious, which means that these are mutations that are actually going to affect you in a bad way. So let's write down the definition of a mutation. You have a flow chart there. And a mutation can occur when DNA's gene sequence of base pairs is changed by either a mistake during DNA replication or by a random chemical reaction that damages the DNA. So then that means that sometimes these mutations are just random. When DNA is replicating during the S phase of interphase, it can just make a mistake. And maybe the mistake is not caught by DNA polymerase. But sometimes these mistakes are due to some type of chemical or some type of exposure. So we know that cigarette smoke damages the DNA. We also know that UV light also damages the DNA, and that's why it's important for you to wear sunblock whenever you're outside. So let's remember that mutations can be harmful. It means that they're bad for you. Mutations can be helpful. That means that whatever the mutation results in gives you an advantage. And the mutations can be silent, which means that they have no effect. So mutations can happen in genes, which are specific segments of DNA, or mutations can happen in the whole chromosome. So let's look at gene mutations. Genes mutations means that you have a replacement of one nucleotide by another. You either add or delete a nucleotide. You can have chromosome mutations, and that means that you have a change in the number of chromosomes. Polyploidy means that you have extra chromosomes, and aneuploidy means that you are missing a chromosome. Let's remember that humans, Homo sapiens, we have 46 chromosomes. Also, chromosome mutations can be due to a change in the structure of the chromosome. So once again, mutations can happen in genes, short segments of DNA, or the mutation can happen in the whole chromosome. So let's look at a moment what point mutations are. So gene mutations can be point mutations, and then these point mutations can lead to other different types of mutations. So let's explore these for a second. So let me get my pen. So what are point mutations? Let's start by defining that. So a point mutation is any time you have a single base substitution. So what does that mean? That means that just one nitrogen base is going to be substituted. Okay? So these point mutations can lead to a frame shift mutation. So for example, let's say that your original DNA has a codon with the nitrogen bases UUA. And let's say that that codon corresponds to the amino acid leucine. Remember last lesson when we were reading the codon table or the codon wheel? We learned how to code for amino acids. So let's say that's your normal amino acid that should be coding. And this amino acid is going to make part of a normal protein that you need in your body. Well, let's just say here that this base gets substituted by the G here. So remember that the way tRNA reads mRNA is by reading a codon. It reads the 3 codons. So now instead of tRNA reading UUA, which corresponds to leucine, it now reads UUG, which corresponds for a different amino acid. And then notice that the whole frame or the other sequences of codons are going to be shifted thereafter. Okay? So then that means that you're going to have a completely different protein, an altered protein, and that may result in some form of altered phenotypic characteristics. What about a silent mutation? So let's look at that for a moment. So in a silent mutation, you still have a single base substitution. So A gets substituted. I'm sorry, not A. Let's go back here. Let me erase that. In this mutation, you have the U here gets substituted by C. Okay? So there is still a substitution, but notice that just like UUA codes for leucine, CUA also codes for leucine. So even though there's been a single base mutation, the mutation still rendered the same amino acid, so there's no change in protein. So you don't even notice that there's been a mutation. And then we have missense mutations. In a missense mutation, what happens? So you are going to code for a completely different amino acid, but perhaps the reading frame doesn't change thereafter. It could, it doesn't have to. So in this particular case, U gets coded by G instead, so there is a substitution of that amino acid. So instead of U, you have G as a substituted I'm sorry, not amino acid, but you have a substituted base, U instead of G instead of U. So now this codon reads valine. Okay, so now you have a different amino acid. So maybe that particular amino acid changes, but maybe the rest of the sequence is not altered completely. And then finally, you have a nonsense mutation. And in a nonsense mutation, the change in the single base leads to a stop codon. So here is the original DNA. So instead of having U in the original codon, you now have A being substituted. So we know that UAA codes for a stop codon. So in these type of mutations, you have a shortened protein or a protein that wasn't fully synthesized or developed. Okay, let's continue. So let's look at some examples of point mutations. Examples include cystic fibrosis, hemophilia, or Tay-Sachs disease. Now let's look at sickle cell anemia for a moment here, which is another important example of a point mutation. Sickle cell anemia results in abnormally shaped red blood cells. The cells are shaped like a crescent or sickle, and they cannot carry enough oxygen. The sickle cells also get stuck in blood vessels, blocking blood flow. This can cause pain and organ damage, and this is a common condition in 1 in 12 African Americans who have the sickle cell trait. So instead of your normal red blood cells looking like little donuts here, they become sickle or crescent shape. If you want to Google it, go ahead and search for CRISPR gene editing therapy for sickle cell anemia, because back in 2019, scientists were able to use this therapy for the first time to treat sickle cell disease, which means to go and replace the mutation. And it seems to be working. I was just reading an article not long ago about how the results have been very promising. So here we see in sickle cell anemia that instead of glutamic acid being made, which is the correct amino acid here, notice that there is a point mutation, and instead of glutamic acid being made here, T is substituted by A, and that changes the amino acid sequence here, and now you have valine. So then that means that this whole sequence of amino acids is going to be different, and then that means that the protein is going to be different, and then that means that the sickle red blood cell is going to appear, because now there is an incorrect set of instructions for making the hemoglobin in this red blood cell. All right, continuing. So there are different types of structural chromosomal mutations. So we're going to learn a few of them right now. So in your note taker, make sure you write down the definition for the different types of chromosome mutations. So let's look at the first one here. The first one is deletion. And in deletion, you have a loss of all or part of a chromosome. So let's pretend that this is your normal chromosome. Okay, and let's pretend that that normal chromosome has gene sequences or gene segments called A, B, C, D, E, F. Okay, so that's just a random alphabetized name we're giving the genes. So in a deletion chromosome mutation, what happens? What do you see happening here? A, B, and you have the segment or the gene C being completely deleted. Because the chromosome looks different, then of course you're going to have different proteins being made. You're going to have an altered phenotypic effect. In duplication, we have an extra copy of all or part of a chromosome. So here you see that the chromosome being altered because you have an extra section of the chromosome that's been duplicated, so 2 Ds. In inversion, you have a reversal in the direction of part of the chromosome. So this part right here, the CDE part, has been reversed. So instead of the normal reading frame, now this section has been reversed and it now reads A, B, E, D, C, F. Okay. In insertion, you have part of the same chromosome from a different part within the chromosome that broke off, and it's attaching to a different location. So notice here we have this section of the chromosome that should be somewhere around here, right? It broke off and then it reattached itself on a different part of the chromosome, again making a mutation. And then you have translocation. This is where part of one chromosome breaks off and attaches to another chromosome. So let's say that this is this is two different chromosomes, and let's say that this is the correct gene sequence. Well, notice how one part of a different chromosome attached to another part of a different chromosome. So it's kind of like a cut and paste here. This section was cut, this section was cut, and then it was pasted on different parts of a different chromosome. So all these are types of structural chromosome mutations. So let's look at actual examples of these types of mutations. So here we have a couple of disorders or diseases that are due to an alteration in the structure of the chromosome. So Huntington's disease is a neurological disease. That's an example of a chromosome mutation. Or muscular dystrophy is also an example of a chromosome mutation. Muscular dystrophy is when your muscles stop working properly, and this is an inherited disorder. And Huntington's disease is where you have a neurological disorder. That means that you have impaired movement, impaired coordination, and it's also an inherited disorder. And then you can also have alterations in the chromosome number. So in our genetics unit, we learned how humans have 46 chromosomes, and anything that is not 46 is an alteration in the chromosome number. It's a mutation, and it can lead to different phenotypic effects. So for example, we learned that in Down syndrome, a person has 47 chromosomes instead of 46. So here you see the extra chromosome in location number 21. So we learned in the genetics unit the different phenotypic effects of someone with Down syndrome or trisomy 21. Klinefelter syndrome, that's when a person has 48 chromosomes, and the extra chromosome is located in the sex chromosomes. So a person's 23rd sex chromosome is XXY. So we learned in the genetics unit that this person has 2 X chromosomes and 1 Y chromosome. So here you have a male with female characteristics. And then we see in Turner syndrome where you have a female, because it only happens in females, with 45 chromosomes, and that's because in the sex chromosome you have a missing X chromosome. So can these mutations be fixed? So there are 2 ways that mutations like these can be fixed, and this is what we call novel gene therapies, and it's a very exciting time that we live in because we see a lot of different techniques that actually go to the root of the problem, which is actually the DNA. Because remember that the DNA is what carries all of the instructions. So in your notes, you are to describe 2 novel ways a mutation can be fixed. And the first one is DNA editing with CRISPR-Cas9. So the CRISPR-Cas9 is a mechanism or complex that works like a biological version of word processing, find and replace. So pretty much what happens here, you find the DNA segment or gene that's damaged. So then scientists are able to design a guide molecule, and we're going to learn more about how CRISPR works in our DNA technology lesson. But scientists are able to design a guide molecule that is able to find that defected or that DNA segment that contains the mistake. And then what the guide molecule does, it leads a DNA cutting enzyme to cut out, kind of like, cut and paste, but you paste it with something that's healthy. It cuts out that defective DNA strand, and then you also introduce a healthy DNA strand. And sometimes this is just by DNA repairing itself and repairing itself correctly, or by introducing a healthy DNA strand that's also guided. So this is the first technique. Another technique of how scientists are now fixing mutations is through vector gene therapy. And this is where cells are removed from a patient, and in a laboratory, a virus is altered so that it cannot reproduce. So you got to remove all the bad parts of the virus. And then what you do is you insert a gene into a virus, and then that virus is going to be mixed in with cells from a patient. And then those cells become genetically altered because now those cells have been infected by the virus that carries the correct DNA sequence. And then you're going to take those cells and you're going to inject them back into the patient with the hope that once these cells are back in the patient, they're now going to work properly and produce the correct desired protein. Of course, this can only happen in cells that are not constantly reproducing mitotically. Because if that's the case, of course, those cells are going to reproduce with an incorrect type of DNA. So that's it for today. Make sure that you can pause the video and finish taking your notes. And I hope you enjoyed today's lesson.